In this video, we'll talk about an important concept in genetics that connects mutations with transmission genetics, and that is the concept of complementation. Let's start with an example. We haven't talked about Mendel's pea plants in much detail yet, but you'll get very familiar with these plants soon. There's this plant that normally has purple flowers, but imagine that you have two mutant lines, both of which have white flowers instead. We also know that these mutations are recessive, meaning that in both of these lines there are two recessive alleles that make the plant produce white flowers instead of purple. Let's say that you obtain these two lines independently and you call these recessive alleles A and B. Because these mutations were generated independently, you don't know if these mutations are in the same gene or not either. To answer that question, we use a complementation test. We cross the two mutant plants and look at the flowers in the progeny. If the progeny flowers are purple, we say that the alleles, or the mutations, complement each other. Something was broken in the A line, but crossing it with the B line fixed it, and vice versa. We also say that the A mutation is not allelic to B. In other words, the two mutations must not be alleles of the same gene. They have to be in different genes. How does this work? Here's how. In line A, both copies of gene A are mutated, but gene B is normal. In line B, both copies of gene B are mutated, but gene A is normal. We normally don't write these things out because we normally write the genotypes of genes that matter most for an organism, usually those that are mutated or different. So, for the A mutant plant, we'd normally write lowercase a, but in reality, we should write lowercase a, and then uppercase B to indicate that both copies of that gene, whatever that gene is, are normal. It's the same for the mutant B plant. We would normally write double lowercase b, but this plant in reality is double lowercase b and double uppercase A. When we cross them, the progeny inherits one mutant copy of a gene, but a functional copy of the other. Because both mutations are recessive, the single normal copy of A provides whatever function gene A normally provides, and the single normal copy of B provides whatever function gene B normally provides. Together, that restores the pathway that produces the purple pigment, and the flowers are purple again. In this case, we say the mutations complement, and A is not allelic to B. Historically, geneticists grouped mutations this way into what they called complementation groups, which helped them map genes into pathways. What does a genetic pathway look like in molecular terms? Let's suppose there is a colorless compound that is processed by the product of gene A, let's say an enzyme, into another colorless intermediate molecule that is processed by the product of gene B, let's say another enzyme, into a purple pigment. When you have both the A and the B genes functioning, the flowers will be purple, of course. Now, in the B mutant line, the function of gene B is missing. So, sure, the product of gene A can turn the colorless compound into the colorless intermediate, but then without the product of gene B, the colorless intermediate would not be turned into a purple pigment and the flower stays white. Similarly, if gene A function is missing, the first colorless compound can't be transformed into the colorless intermediate, so even though gene B is still functioning, it's lacking its substrate and it cannot produce the purple pigment. Now, when the progeny plant is heterozygote for both genes, there will be one functioning copy of each. Now, the first colorless compound can be turned into the colorless intermediate by the one functioning copy of gene A, and the colorless intermediate can be turned into the purple pigment by the one functioning copy of gene B. Now, let's consider a different case. Let's suppose that we have two mutant lines again. This time we have line A and line W. This time, when we cross them, the progeny still produces white flowers. We say that the mutations do not complement. What's happening here is that both mutations affect the same gene. Let's call this gene the flower color gene or FC gene. When mutations do not complement, what usually happens is that line A carries a mutation in the FC gene and line W carries a different mutation of the same gene. When crossed, the progeny inherits one chromosome with a mutant A allele and another chromosome with a mutant W allele. Neither mutation is present in two copies, but both copies affect the same FC gene and make it not functional, so the flowers remain white. In this case, we say W is allelic to A. They're different mutant alleles of the same gene. For many decades, complementation tests were a central tool in genetics. By systematically crossing mutants and seeing whether they complement or not, scientists were able to build complementation groups and map genes into pathways and later into networks that underlie specific traits. All right, I hope you found this useful, and I'll see you in the next video.